All right. So welcome everybody. Thank you so much for coming here. Just to let you know, this is a session for how to learn bioinformatics. If you're here for something else, then enjoy the food. All right. Okay, so I figured, we need to outline the goals for this lecture per se. I do this for all my lectures. So the goal for this particular intro session would be twofold. One would be to understand what bioinformatics entails. So at the very least, I want you to leave with some basic idea of what is bioinformatics, the definitions, and the difference between some of the commonly used terms for or synonyms for bioinformatics. And then also I'll be happy to give you some resources on how you can learn bioinformatics, including we'll talk about some of the stuff that we're offering here at the Cancer Center, but there's also online resources which you can use. All right. So at the very least, you'll learn two things today. Right? Okay. So, well, who here does next generation sequencing, or has done it in the past, or is planning to? Cool. So, as expected, most of you have done some NGS or are planning to do some sequencing in the future. So in that case, you are in the right place. Let's go on a little journey today then. All right? So if you are planning on doing sequencing, this was a paper that was published in 2016. And it's a really interesting read. It just talks about where the real cost of sequencing is. And it used to be that sequencing was very expensive, and it used to be that the actual library prep, the materials, all of those were the actual cost of sequencing. But now we've seen that the real cost of sequencing is just the computation that is now taking place post-sequencing. And that seems to be the biggest bottleneck in sequencing as well. Because you have done your analysis, you have done your sequencing, your prep, everything is ready, and now you have to wait for someone like me to give you the answers. And if you're impatient, again like me, then you want to know the answers yesterday, not tomorrow. So that was something that we've seen. So with more and more data, so in this paper they talked about that how they in 2016 they had 4 petabytes of data in SRA, which is a sequence read archive, which was started in 2007. So just in about 9 years, they had 4 petabytes. Who here knows what a petabyte is? Or how many zeros it has? So that's 10 to the power 15. So basically it's a lot of data, essentially. And half of that is public. So you could technically go to download 2 petabytes of data today. You won't have the storage. But you can download it technically and start analyzing and do a lot of fun stuff with it. But to say the least, it's overwhelming. So if you even start with terabyte of data, or forget terabytes, like 10 gigs, which are, these days normal sequencing runs can run up anywhere between 5 to 10 gigs, just the files themselves for one sample. You can see how it can be overwhelming, and the computational speeds and the parallel processing that you need seriously, it's a lot. Not to say the knowledge that you need just to process that data. So I'm here to tell you that it's okay to be scared if you are. And that it's perfectly normal, but there is a way out. So there is a way out, and today we're going to talk about some of the solutions. What are the ways out from this kind of a problem? And that brings me to who am I? Well, so my name is Vishal, and basically I'm a computational biologist turned bioinformatician now. I work with Ting Lab, and sometimes I also work with Haber and Maheshwar Lab. But I'm mostly working for the Ting Lab and the MGH Cancer Center. That's why I'm here. Now, so before we, so let's talk a little bit about the first goal that I'd outlined. So has anybody heard these terms? Actually, I just talked about computational biologist. You may have heard I'm a biostatistician. Some people say that I'm a biostatistician. So what is the difference between bioinformatics, computational biology, and biostats? Anyone want to take a guess? Have you heard these terms? Right? So usually they're used synonymously, saying, oh, yeah, one means the other, but they're actually quite different. So So a bioinformatics person, or the science of bioinformatics, is primarily focused on computer-aided analysis of data generated by biologists. So this is the one that is most used, and this is the one that we are going to be working with, and the term that you guys should be most familiar with. So what this does is it uses tools, programs, and existing software, right, to do your analysis. Computational biology, on the other hand, involves the attempt to write such programs, tools, and to come up with biological models in silico. So basically creating, writing programs and models and software. So that's comp bio. All right? We won't be covering this. This is not what we're going to be doing, because that involves a very high degree of programming, which frankly you don't need. And then biostats entails the development and application of statistical methods. So someone with a master's in statistics or biostats would probably be more interested in how to model data differently, and what is how one type of data might be different from the other. How is microarray data different from sequencing data? All right, so those kind of questions. Right. So we've met goal number one. Now you understand what's the difference between bioinformatics and some of the other synonyms. So goal number two. All right. So the goal number two is what are the resources that are available to learn bioinformatics? And a quick Google search will pull up all of these courses that are now available. There's one on edX, there's one on Harvard Extension, there's on Coursera. So essentially there's lots and lots of online courses. And frankly, I find that they are good. They're actually very interesting as well. And you know, technically anybody should be able to do this. So with that, we end the talk, because both the goals are met. Just kidding. All right. There is one problem, and that's the problem why you guys are here. The problem is that there's a steep learning curve. What that means is that basically I myself have tried to take some of the online courses, and almost always, you know, week one goes by, I do a little bit, week two comes by, fall behind. By the time it's, you know, end of the month, it's, you know, 6 weeks in, I have pretty much lost all touch with the class, and the class has moved on. And because it's more self-motivated, which I'm not, so what ends up happening is that I will end up giving up on these courses, and that's the end of it. That brings me to the next part, which is, let's talk a little bit about a story. So it started in 2014. I had been working at the Cancer Center now for 2 years, and I was doing some analysis for postdocs, and I noticed certain patterns in those discussions that I was having with technicians and postdocs, which was that I would spend a lot of time explaining the computational aspects behind the sequencing. So they'll ask me, why this aligner? What is the QC? What is QC? What did you do? Why have you, taken my reads and cut off the ends? And those kind of fundamental things that basically I was interested in explaining. So in the end, it would be a half-an-hour meeting, and I'd spend 2 hours just talking about this other stuff, which is fine. I also noticed that I really enjoy this thing, so I felt very good that I was imparting some knowledge. It's just that, you know, eventually as the projects grew, I got less and less time, and then I said, well, maybe there's an opportunity here. So I did some research, like any good researcher. Here's my favorite scientist and researcher. I did some research and I found some facts. So this is in case you don't know, this is Dwight from Office. The guy is a wonderful researcher. He has lots of facts about everything. So here are some facts for you guys. So fact, 24% of all new MDs will have some prior training in computer science. So biologists and doctors need to understand and perform data analysis. And this is not just statistics. This is gonna more and more, this is becoming a reality, which you guys have found as well. So another thing, so a lot of courses out there would be, you know, everyone's in a hurry. They want to do everything in 1 day, or 2 days, or a week tops. They'll be like, you know, become a bioinformatician and take our course in 5 days. Before here, I was at Cold Spring Harbor where I did my postdoc, and I found that they had a course for bioinformatics. As a person who has a PhD in computer science, I decided, oh, this should be easy. I took that course. 5 days later, I was lost. And this is after having a PhD in computer science. I figured that what just happened, because they throw so much material at you, you can't keep up. All right. So the second part, which is that repetition project-based learning is long-term. So all students learn at a different pace, and that's something to, average time spent per course is 5 days. Average time required for a course is about 30 days. So there's a gap there. Then there's also the last one, which is the financial aspect of learning anything bioinformatics related, which we've seen that there's a 16% increase in jobs per year, every year. It's not cumulative. Every year there's a 16% increase in job for bioinformatics and computer science. So there's no downside to this aspect, right, if you've learned bioinformatics. All right. Okay, and then this is just a graph of faculty hires in 51 universities. And you can see this is for computer science, but it's also, there's a strong overlap with bioinformatics. There's a strong correlation there. So it basically shows that, if you're interested in academia or industry, either ways, some prior knowledge of computer science or bioinformatics will be good for you. All right, so I started a course. I started this course around that time in 2014. And it started out with, I think, 3 or 4 students. And nowadays we've had semesters where we've had 20 students and we had to turn people away as well. Okay, so I started one course which was basically in bioinformatics, and then it used to be around 8 to 10 weeks long, where people would just come in and we talk about bioinformatics, lecture, practical format, and then it grew into 2 courses because there was a demand for people who wanted to just still have a one-day sort of refresher or go through something where you have the same bioinformatics things, but it would be online. So that was the second course I started. And then now, this semester, we're starting another course, and that's the third course. This one, and we'll talk in detail about these as well, so don't worry, not just skip this. So there's a third course we're taking, so this is for people who have had experience with one of these, at least one. Ideally, the first one. So you've learned some bioinformatics, you know a little R, you know a little bit of programming, you know a lot about sequencing. Enough that you can then go into a little more advanced learning for single-cell RNA-seq. And the concepts that we'll talk in a bit, you will see why they are advanced. Okay, so this is our team. There's me, and the very first person who was to join our team was Dr. Ting, David Ting. With his blessing, we were able to start this course and officialize it. Sitting right there. And then Dr. Ben Whitner, PhD in mathematics, also a bioinformatician. He's written some packages in R and talks about statistics with us. Kendall, he's a postdoc in Martin R.E.'s lab downstairs in pathology. Also very smart, wonderful guy, does a lot of chip-seq analysis. He does that lecture. And this semester we'll be joined by Dr. Vajira Virasekara, who we've been working all summer trying to create the third course for you guys, the single-cell RNA-seq course, which he'll be leading. So we'll learn more about that, but before that, over to Dr. Ting for a few minutes of his stuff.
All right, so thanks everybody. So this course is really, as to just reiterate, some of what Michelle was saying, is really trying to educate all of you who are not in the computational space how to at least start to look at your data. We don't expect you to become computational experts. It's more about how can you communicate with our computational colleagues so that there's less inefficiency. And part of that talk that I will give in the course will be on sequencing technologies, and this has evolved over time since I've been here as a postdoc and now faculty. And so, everyone thinks that there's just a Illumina sequencer, but in fact there's actually a lot of options that people are not necessarily aware of. And those are the things that I'm going to talk about, are what are the different types of sequencers, what are the different types of library construction, what are the things that you think about as you sort of actually do your experiment. So most people just say, oh, just send to the sequencing core, and they'll give me all the answers that I need, but that's unfortunately not how it usually works. So, all right, so so some of the things that you should be aware of, we do have a HiSeq 2500 at our disposal. It's part of the core facility in the MGH molecular biology department at Simkes. The Cancer Center did pay for part of that, so we do have quote priority access to that. This is the older sort of like high throughput type of sequencer that a lot of people are aware of. We have 2 MySeqs in the Cancer Center here in Charlestown. We also have a NextSeq, which is not on here. There is a PacBio system nearby in UMass that Howard Hughes investigators have access to. Daniel Haber is a Howard Hughes investigator and does have access to that as well. There's a couple others like Oxford Nanopore and the 10X platform, which are here in the Cancer Center, and certainly we'll go over that in more detail at the talk. So one of the biggest things I ask people is like, what is the question you have, and there's maybe a different sequencer that's required to answer your question. Another thing is what do you have to think about when you do library construction? Like some people just say, oh, I just want RNA-seq, but there's like total RNA-seq, there's mRNA-seq, there's small RNA-seq, microRNA-seq. So there are a lot of these like little nuances that people don't think about. And the last thing is caring about sequencing quality scores, which there's an example that I discussed in the class in detail that this is something that you may not think about because you just assume that the ACTMG is actually ACTMG, but there is an error rate, and so some people make calls on mutations that are just errors. And so this is sort of a global view of the different sequencers. I'm not going to go into this in deep detail here, but just to sort of say like different ones have different error rates per base. This sequencer is actually now gone. Helicose is a single molecule sequencer with high error rates. It still exists as a company called Sequel. We do have one of those sequencers here. Probably only 6 left on the planet. Ion Torrent, we do have one in the Cancer Center. No one actually uses it. This is the workhorse that Thermo Fisher sells. PacBio, like I said, is now owned by Illumina, and then Oxford Nanopore. So, there are different types of read length, different amount of DNA input, amplification bias. We'll go into these things in detail, in the class. And I just sort of put this up here as an example of like, if you're looking at mutation detection, unfortunately ABI had the best error rates. It was the lowest, but that platform died. So Illumina is now the established platform. It has a 0.1% error rate, so that means one base out of 1,000 is incorrect. So you need appropriate depth. That's why people talk about depth all the time. I need 100x coverage, I need 1,000x coverage, because they really want to make sure that what they see is not just quote an error. RNA splice variants, there's some limitations with Illumina here. They do have long read, pair end reads. They did buy PacBio, so they have this capability of now looking at splice variants and translocations. They're looking at novel RNA discovery. This is really a combination of PacBio or total RNA-seq and Illumina, which is different than mRNA-seq. The vast majority of TCGA and ICGC is all mRNA-seq. So people are like, oh, I'm going to mine those giant databases. They've spent hundreds of millions of dollars on it. Guess what? All of it is mRNA-seq. So you can't find long non-codings or anything like that in those data sets. In fact, we probably have to redo all those sequencings in a total RNA-seq fashion. Repetitive sequences is like my personal favorite. This is what my lab does. I won't go into detail on this, but certain sequencers, i.e. the Illumina, can't see repetitive sequences well, so that's why we cannot actually sequence the centromere very well. If you look at our genome alignments, the centromere is like a black hole, but maybe the centromere is actually fairly important. And if you have very small amounts of material, sort of one of the things that I did as a postdoc was develop single-cell RNA-seq before we had all these 10x platforms. And so you can do things that we couldn't do before with these amplification techniques, but there are some benefits of using platforms that do not use amplification to create bias, like the single molecule Helicose or PacBio or Oxford Nanopore. And this can give you different types of readouts that you can't get with Illumina. And these are just sort of examples of questions of why we care. total RNA-seq, mRNA-seq, there are differences, obviously. Stranded RNA-seq, why would I care about what strand? Well, there's a lot of evidence indicating that antisense transcription is important. That's how hot air was found, which is one of the long non-coding RNAs in the Hox gene locus. Pretty much we only do pair end at this point. I don't think many people do single end. It's a cost thing. Quality scores, again, this is very important when you're calling single nucleotide variants or SNVs or mutation calls. People sometimes don't realize that there are differences on the sequence read, where the read is and what kind of error you get. And so one of the things that we see is that if you go from base 1 to 50, usually the quality scores are pretty good. But once you're past 50 to 60, 70, the sequencing quality starts to drift. And if you're past 80 to 90, you start to see higher error rates. And so if you see all these aberrations occurring in your sequencing, it may be because you're at the end of your sequencing run. I'll sort of unfortunately highlight an example of that where it didn't end up too well. So here's an example. So this Fred quality score is what people talk about. They typically want a Q score of greater than 30. So this is a base call accuracy of 99.9%. Like, wow, that's pretty good. That's like, really, really accurate. I mean, it's like an A plus in most places. But when you think about this, there's 3 billion base pairs in the human genome. And so this is an error rate of 1 in 1,000. So if you just sequence, you will find errors 1 in 1,000 base pairs. That's a lot of mutations that you found. So that is the average quality score you see in Illumina. Now this is the beginning of the sequence run and the end of a sequence of a read. And this is around 100 base pairs out here. And so what you can see is basically green is the Fred score, good, yellow is intermediate, red is just terrible. And what you can see is this sample, we got a pretty good quality score. The same sample on the same run, sequenced, done with a different library prep, had terrible error rates at the end. And so if you didn't know that you had all these potential errors, or did quality control checks, or did filtering of these reads or trimming of the reads, you would see a bunch of errors in this sample and not this sample. So if this was the cancer genome and this is the normal genome and your paired biopsy, you would see a bunch of mutations. So, one example of this, and this is, a very interesting paper, and I think that there's, again, I'm not trying to sort of say that this was a bad paper, but what they were looking for were RNA and DNA sequence differences in the human transcriptome. So what they did is they took RNA and DNA from the same sample and sequenced them. And then they said, I'm going to look for differences in the RNA compared to the DNA, because those are going to represent RNA editing events. Okay? So that sounds great. So they did. They were like, wow, we see thousands of these events, all these A to G, T to C. RNA editing is amazing. RDD is a significant thing. Science paper. So the cool thing is that they did show that these events can cause actual protein coding events. So you can actually have a mutated protein that is an RNA mutated species, but the DNA would be intact. So you can actually have a mutated, like theoretically, KRAS, but no mutant KRAS in the DNA, theoretically. And so they proved this for one of these examples. And so I do think that this phenomenon is real, and I don't think there's any. I think other people believe this as well. And so, this was a really cool finding and published in Science. However, the problem is when you're in science, there's a bunch of other people out there who like to look at your data and maybe try to discredit it. And there are 3 independent groups that had found that all these mismatches they had were maybe not true. And so this is what they showed in their I guess, response or sort of query, is that when they looked at their raw data, all the events were occurring at the end of the sequencing run. So the sequencing here went from here to here, or here to here, and they saw all these events occurring at the end of the sequencing. So now that I've told you, very end of the sequencing, you have higher error rates. Unfortunately, they found that the vast majority of these RNA DNA differences were in fact sequencing errors at the end of the sequencing. So I tell people, it's a great science paper, but you kind of just don't want to be that close to it. So this is why it's important to be aware of, what sequencers do, what are the potential issues or false positives that can occur, so that you don't come into this situation. And certainly I don't think the science is bad, it's just, these things happen, and we just hope that that isn't you. So other things I'm going to summarize is in our talk that I'll give is, sequencing library construction. People think it's like a complicated thing, but it's actually fairly straightforward. I have consistently hired techs in my lab who are trained within a couple weeks and can do these things. So postdocs, grad students, technicians, even undergrads. It's not a scary thing. It's basically similar to setting up a PCR, but you're doing many more steps to it. Again, a lot of people don't think about, questions. So what is the question you need to be answered? This is very important because this will affect which construction kit, which sequencing depth, what sequencing platform. And you want to do that because at the end of the day, if you make a bunch of data and it doesn't help you answer your question, it's going to waste a lot of your time, and unfortunately, that's not a luxury that many of us have. So the other thing I tell people is you need to confirm sequencing findings with some kind of alternative assays to exclude technical artifacts, and this is, I think, something that is not universally done, but I think again with people's history of having some publications have difficulty because no one can replicate, this is an important thing. And the very last summary point before I leave is really, so everyone says, oh, okay, if I just do enough, DNA-seq, RNA-seq, ChIP-seq, DRIP-seq, IP-seq, whatever-seq, and I make a large enough Venn diagram, it will tell me what I need to do for my Science or Nature paper. That's never true. So the one thing I tell people is sequencing provides you all this data. As many people in my lab understand, because they've been working with me, it gives you all the answers to everything. And all we can do in computational biology is give you statistics. And pretty much every time you ask to do statistics on a large number of data, you will always find stuff. You will find hundreds and hundreds of potential things. And you can go validate every target, and that's probably going to be, I would say a minimum 300, maybe 100. And that's just not a great path. So I always tell people, when you do this, you have to go in with some hypotheses. Like, just have some hypotheses, because GSEA, gene ontology, and PEG are not gonna give you the answer. And if they do, it probably was already published somewhere, because that's why they're in there. So, you're here to find new stuff, and so you have to sort of be creative about having questions and then looking at the data for the answers, rather than hoping that the answers tell you, how to write your paper. Because if you do that, I've seen this time and time again, you will keep looking at the data like looking at the stars in the sky and wondering what it all means. And that's all I have to say. Any questions before I leave about anything?
So, just a general question. In your lab, you don't go like hypothesis free? I mean, like, let's say I started with treating with a condition and want to know whether it stimulates a specific pathway.
Yeah, so you know, if you have a testable system, I think like you can do the experiment and see what comes out. But what I almost universally find is like, you know, you get like all these Go terms, the computational biologist like, I've given you all these p-values. This is all I can do. Like he or she can't tell you what's true, and so you actually have to make sense of all those Go terms and write a story about it. And just putting them out there is not sufficient, because what does that mean? You know, that's like, that's the thing, what does it mean? So that's why it's always easier to have a hypothesis. Now, when you think about how we do these GSCA and Go and gene set kinds of things, you know, they're based on gene sets that someone put together. It wasn't like, you know, we had a meeting with 3,000 scientists and we all took votes on the genes that should be in Go term, you know, intracellular pathway. I mean, what does that mean? Like, intracellular pathway, you know, like, it's in the cell, great. Like, not helpful, right? So there will be a p-value for that, and you can write about it. It's, you know, I think that's the difficulty. But if you go in instead with like, you know, I really think that I think that this EMT thing may be important. Then you actually have a hypothesis. You look at the EMT genes. And actually, your false discovery rate goes down because the number of hypotheses is now 1 instead of, you know, basically 20,000. Does that make sense? So, any other questions about sequencing infrastructure or anything like that? We'll certainly go over, you know, a lot of these things in the talk. But, all right, I'll give it back to the chief.
Thank you, Dr. Pink. Yeah, thanks again. So let's just go back to that example one last time just to go over it. Basically, why did that paper get published? And you know, it was probably because the bioinformatician had like 20 projects, and he just gave them the data, and I was like, here you go, you can make sense of it, and then he moved on to the next project, right? But guess what? The postdoc, it's not really his fault or whoever was the first author on that paper, and the law. They're basically be like, oh, look, I've got this data and this makes sense, like, you know. And so they go ahead and, you know, find a few findings that confirm, and obviously they're not going to test all the candidates, they test a few and they're like, oh, it makes sense. And so the paper got published. But then when the rebuttal came, you know, and that was probably people who wanted to take this group down for whatever reasons, and they did their analysis, they went into in-depth. Now the postdoc is coming back to the bioinformatician and saying like, hey, this happened, and now I'm the one who has to answer. So that's the kind of situation that we're trying to teach you to avoid. Basically, because yes, you will still rely on bioinformaticians a little bit. I mean, some people though, I would say about 25 to 45% of the people who take this course end up doing their own analysis. A lot of times people will just go back to their comfort level where they're like, okay, I want to still work with a bioinformatician, but the interactions have, time and time again, my fellow bioinformaticians have come back and told me, it's like, well, thank you so much for teaching so-and-so postdoc about bioinformatics, because now I can actually have a conversation with them and I don't have to explain everything. They pretty much know everything I'm talking about, right? So that's the other aspect of this course that, you know, a lot of people can gain from, which is that you don't really have to feel like you don't know what to ask or what the bioinformatician is saying, because sometimes you feel shy, you're like, oh, should I ask this question? Well, that's the point of the course, is you can ask all the questions to me first, that way you don't have to worry about them later. All right, so let's get to the basics, what's covered. First, the long course, the one that is. So first of all, let me just get this out straight away. Except for the third course, which is later, in this particular one, there's no assumption. I don't assume that you even know how to, you know, do anything on your computer apart from opening a web browser, which sort of is a valid assumption nowadays. But I don't assume that you know any programming. I don't know you even know what these terms are. I don't assume that you have to know anything about anything in terms of computer programming. All right. So if it sounds like you are still conflicted, don't worry. We'll go through every single thing, all right, that is required to get you to be able to do your analysis. All right, so we talk about Unix, which is, you know, an operating system that most clusters work on. So we also talk about what is a cluster, why does it need Unix, what is the history behind that. Pretty much you guys understand that you get sequencing data, right? So what is the data format? What are the different data formats? How are the genomes out there? What is their format for that data? What is the difference between a GTF, GFF, FASTA files, FASTQ files. Maybe you've encountered these terms and wonder or not, but we go over that. We talk about quality control. We talk about sequence alignment. How do you align which aligner? What commands to use? How do you run that in parallel on a cluster in the Unix operating system? Believe it or not, you will be able to do this once you take the course. It might sound like a lot right now, but that's the aim. That's why we do it slowly. How do you count reads? How do you make a matrix of your expression matrix, as we call it, where you have your genes on the rows and then the samples on the columns? And that matrix, how do we take it and take it into R? R is a programming language which you use for data analysis. It's like basically it's a much more sophisticated and easier version of Excel. Well, I wouldn't say it's easier, but it's definitely a lot more, there's a lot more features. So what is R programming? We go over that over 4 or 5 lectures. 5 lectures. We talk about variables, loops, functions, data manipulations, graphics. So you will be able to make graphs like this by the end of this class. And then, of course, we talk about differential expression. What is normalization? And how can you go from differentially expressed genes to representing them like this, which is what basically publication-worthy figures and heat maps. And then, so Ben is the Dr. Whitner. He's the one who wrote this package, and he'll come and talk about that. But I also use this package myself, so I have a bit of knowledge about this as well. Ben also talks about statistics, which again I also cover a lot of statistics as I'm teaching the course, because I can. But for deeper concepts, Ben comes and gives a talk about null hypothesis, distribution of data, testing, hypothesis testing, what all that stuff is, statistical power, pretty much most of the stuff that you need to know. Towards the end of the course, once we are we have our differentially expressed genes, or list, or whatever, just a gene set, we go through GSEA, or gene set enrichment, what are the pathway analysis, we talk about genome browsers, how we can, look at this data, visualize it in the browser format. But that's not it. The course doesn't end here. It used to end here, but it doesn't end here now. We have added some extra lectures here. We talk about variant and mutation calling, and that's done with Kendall. And then we also talk about chip-seq analysis. Now, this chip-seq analysis is one lecture. We're thinking about making this into a separate thing, but for now, it is going to be offered as part of this course as well. If people are doing more and more chip-seq and ATAC-seq, you can bring in your questions, and we'll go through this, and we also do a practical and teach you from start to finish how you can do that. All right. Apart from that, finally, I give you, and this is probably the biggest perk of this course, actually not the biggest perk, one of the perks of this course is I give you my pipeline, and I teach you how to edit this pipeline using Python and Snakebit. Right. Now, with R programming, you will have some basic knowledge of programming. I will translate that knowledge and take concepts from Python and show you how you can use a pipeline, run it. It's all running right now, and you'll be able to do this. This is pretty much the last, actually the second last lecture. So, is that it? Well, not really. There's a cherry on top. We also talk about there's machine learning basics. So this is a lecture that I added towards the end because people want to know a lot about what are the fundamentals of machine learning. What is principal components? What is an SVM, support vector machine, random forest? What are neural networks? So this is just basically an introductory overview lecture, right? So we go through that towards the end of the course. And so this is the summary of the long course. So there are about 15 lectures, give or take. It could be 14 or 15. I usually keep 15 because sometimes I don't rush, like I'm talking now. I don't rush. Because the reason for that is I like to make sure that everyone in the class, we keep the class size short, which is why people, when they are done understanding everything, that's when we move forward, right? So if you're not on board, we're not going to be like, okay, I don't care, next lecture. No, we're going to go over that stuff again in the next lecture, supposing you don't go over. There are 14 practical sessions with these lectures on the day of the lectures. So whatever we cover in the lecture, we also translate that into a practical. So you sit down there with your laptops, and we'll work through that on the cluster or in R. Then there is one midterm, and then there is one final exam as well. Again, these are all very informal. They're not as scary as it sounds. It's just a midterm. You have a lot of time to finish it. There's no deadline for it, but that's basically how you learn through this course. Is once you have that separate exercise, you do it on your own. That's when you really start to understand things. All right, same with the final. There are review sessions every week as well. Actually, most weeks. I shouldn't say every week. So the weeks where we are not able to get the most out of the practical, or someone still has some doubts, they come in the next day, and that's where we cover the review session. Apart from that, I also have sometimes you can arrange a one-on-one time with me as well, right? And then you also have 8 homework assignments that are part of this this course. So guess what? We also have all of this stuff online. So just like we're recording this session today, we record all of our sessions, which means that if you're traveling for a conference or if you're going somewhere, you have a meeting, which happens a lot, by the way, you can go online, and there's already existing more than 30 hours, but as you go along through the course, we also have the newer versions of the same material come in every week, so you can listen to exactly what happens while you're away, along with what you need to finish before your next class, right? So all of that is available online as well. All right, so the shorter version of this course, this is essentially a one-day course. This was started because I got some demand for it. Frankly, again, when I started this lecture, I mentioned that how I'm not in favor of putting a lot of information in one day, but some people can handle it, so I figured why not? We'll create one-day course where we're going to do everything online, all right? So here, the idea is you can understand the theory, the concepts, and I can give you some online tools which you can do for light analysis. By light, I mean that your file sizes are less than 2 gigs. You're not having big data, you don't need to run it in parallel, and you are not in a time crunch. Because if you're in a time crunch, forget it online. You can't finish your analysis in a week online. It's very hard because it runs serially. Do people understand serial and parallel? Right? When I say serial, it means you first do analysis A, then you do B, and then you do C. Whereas when I say parallel, you do A, B, and C at the same time in parallel. So on a cluster, you can run parallel analysis, like in the cluster, which is what we learned in the long course. And the data size is not a limit. Whereas in this, the data size is a limit, and so we'll go over that in this course, how to work with smaller data sets online. And pretty much we talk about the same things that we talk about in the long course, just basically in one full day. So it's an action-packed day. And then we talk about these things to web-based tools. For example, we use GenePattern and Galaxy. These are the 2 tools that we introduce in that course, and show you how you can perform online alignments, online heat maps. Same thing, we talk about statistical concepts. So there is an overlap between these 2 courses. If you're taking the long course, this would be a waste. If you cannot take the long course for whatever reason, this would still give you something to work off of. Okay, that's my 2 cents. In terms of preference, I would always prefer to have the longer course because it's spread out over time. But again, as I said, some of us really want everything compact, and that this is a perfect solution for this kind of problem if you're having. All right. So we talk about GSCA, pathway analysis, and data analysis from different online sources. Oops, sorry. Okay, and we also go about variant calling and ChIP-seq and machine learning. Okay. Sorry, so machine learning as well is covered in this one day. So like I said, it's a full day. We started around 8 a.m. We ended about 6 p.m. So that's, with about 1 hour break in the middle. So that's one action-packed day where we cover pretty much everything that we can do online with Bioinformatics. Okay. All right, so now about single-cell RNA-seq. So if you are at the point in your bioinformatics journey where you're already able to do some R programming, if you already understand some basic concepts about what is principal components, you know what heat maps are, you can do a little bit of programming in R. By little bit, I mean you can load packages, you've done some differential expression using DE-seq or some other EdgeR or some other, if what packages I'm talking make sense to you already, then you should consider taking this course, which is, we're launching for the first time. Vajira Veerasekara, who is a postdoc in Nabil Bardis's lab, has been working with me. We've been working together for the entire summer, and we've come up with the material for this. So this is the pilot version of this course. And over to Dr. Veerasekara, who's going to tell you a little bit about this.
Thank you, Vishal. Hi, everybody. So I'm postdoc at Nabil's lab at all Cintas, and a little bit of myself. I'm a biochemist by training. I did my PhD in Utah, where there's like a lot of outdoor stuff to do. So I had a lot of hobbies, and I moved to Boston where it's like none. So I found my new hobby. So I found art as my new hobby. So that's how I became interested in bioinformatics. But I still do a lot of it. I mean, I do wet lab stuff, but I like art. So and the hobby turned into a passion, and like I'm actually pretty serious about it now. So that which means like anybody can. I mean, not a computation biologist but like you can at least be self-sufficient about analyzing your own data, and so you don't have to be scared about that. So a little bit of this course is we've seen like a lot of labs, including our lab. We do single cell RNA-seq a lot, especially when you have organoids, when you have PDXs, you have treatments because you are studying of certain kind of resistant programs. So a lot of labs do 10x genomics or some sort of a single cell RNA-seq. And the reason that you do is because you use bulk RNA-seq, you just see an average expression of the whole, whatever the cell types that you have in that particular PDX or the organoids. But these are heterogeneous samples. But when you do single cell, you can actually get a cell population, and you can say if you have a treatment, and if you are studying a resistance to a particular treatment, you can actually see the shifts of these certain populations, and you can find out, oh, this is the cell population that gives resistance, especially in the context of immunotherapy, which the tumor microenvironment and the ligands that they secrete, the interleukins matters a lot. So that's why we thought like this course would be interesting to offer. So it's basically a 5-week course, and it's a 1 hour lecture or a 1 hour practical. So Siang here, my friend from Leaf's lab, would cover the sample prep using the 10x, how to use the 10x genomic sample prep. Then we'll start covering the alignment and generation of count matrix, and then after that it will be all R. Like, it will be really fun. So then we will use R programming language to do the filtering, the linear and dimensional reduction, and the nonlinear dimensional reduction, which sounds like weird, but trust me, I have the same issues, but it's pretty interesting. So, but at the end of the course, you should be able to know the basic idea of sample preparation and which factors to consider when you prepare your samples, because sample preparation is very important when it comes to these technologies. And most importantly, rather than just pressing enter and just changing the file names of the script, you will understand the principles behind the code that we use, or the statistical models that we use, why we use this, why we need to use this, why we have a linear dimension reduction, why do we use a nonlinear dimension reduction, and how you get those beautiful t-SNP plots, how you sort of incorporate your mutation and the drug response into a single plot, all this cool stuff. And then at the end of the day, the goal is, because these pipelines keep changing, so the single cell RNA-seq pipeline which I learned was Surat 1, I think version 1, now it's like version 3. So these pipelines keep changing, but the principles don't change. So if you learn one pipeline, if you learn the principles behind a pipeline, then in like next 5 years or like after 5 years, there's a new pipeline, you should be able to adapt yourself to this technology. So that's the basic idea that we want to give, that's what we want to deliver through this course, that you'll know how to adapt yourself. So that's all for this course. You can ask more questions if you have.
We cover analysis pipelines like the Sera and ScanCon.
Yeah, so we'll do Sera. That's the pipeline that we would cover in this course. Yeah, and it's so this has like a lot of online resources, especially the Surat, the lab who introduced Surat. They keep continuously updating their pipeline, which is awesome. So the idea is to, after you do follow this, you will know to use the new updates, because it will be keep updating, the statistical model will be keep updating. So we'll be focusing on teaching the principles behind this pipeline, so you'll be able to adapt to the new versions. Okay, thank you, Bishop.
Thank you. Well, that's a brief introduction to this. So as Vajira pointed out, we're basically going to be covering this with the point of 10x sequencing, right? So that's our primary analysis, and we will be following a lot from the Surat manual as well as that. So that's pretty much our guide reference so far, but we will be adapting as we go along through this course. Later on in the next iteration, as has happened with previous courses, we try to include a lot more stuff, which is why so all right, so let's talk a little bit about some of my favorite. People have heard this. MasterCard has these commercials where you say next seek kit is $2,600, my seek $1,125. Doing your own bioinformatics analysis is priceless. But at the same time, it's we live in a material world. So here is the tuition for these courses. Pretty much 100% of this goes to the Cancer Center, which is again used for the development of programs like this one, and to provide resources to people. So, for example, the semester course, if you're outside of the Cancer Center, those are the rates. If you're in the Cancer Center, it's about $1,500, $1,495. For the one-day course, it's about $495. I'm just assuming Cancer Center, and if you're not, we can talk about the other rates and how we can go through payments for that. For this new course that we're launching, we're going to be pricing it at $995 for Cancer Center students, but because it's the first time we're doing this, it's 100% likely that we'll be giving a discount on this for people who registered for this particular course. Okay, so it's not going to be this price for sure. It's going to be less than that. All right. Okay, so you can attend these courses in person or by video. We highly recommend in person. But as I said, you can take it offline and take it on your own as well. One of the biggest benefits of this, and a lot of people use this, is that during the portion of this course and afterwards, I offer free consulting. So essentially, you pay for my time by taking this course once, and then you can come back and, if you're still working on the same materials or same problems related to the course or your own data sets, I'll help you through it. I'm not going to do your analysis, but I will guide you through the analysis, because that's the point. I want you to be able to do it yourself. And people have come back, and I've helped them through their analysis. All right, that's part of it. And then, all right, what's next? Let's to wrap up. Well, the next session starts for September 10th, which is next week on Tuesday, for the semester or the long course. All right, so you have about 1 week to register for that course. The class size is 15. As of now, 12 slots are open. So if you're interested, I'd highly recommend sending me an email today or tomorrow. I do anticipate these slots to be filled up. I'm not going to take more than 15 students, just because that's the optimal number that I've realized where everybody wins. If you have less students, then the interaction is much better. So the one-day course, the classes would be 1 day a week, which is Tuesday. Wednesday, which is the revision session, is optional. So the Tuesday class is part of the course mandatory, where you come in and we do 1 hour lecture, 1 hour practical, and it's from 2 to 4 p.m. All right? So you may have to work with your schedule to free up that time if it's not available. And then the Wednesdays are revision sessions, and that's some Wednesdays depending on that. And then the one-day course is going to be on November 9th, which is still tentative, but that's the date I've booked based on the availability of conference rooms and locations. I will finalize this, so if you're interested in this, again, send me an email. The class size is 15, so there's lots of slots in this one. I do not anticipate this to fill up, but please, definitely. And then the single-cell RNA-seq, this is going to be on Wednesday on October 2nd and to October 30th, so that's 5 weeks, 5 Wednesdays. And this one, the timings are fixed, and it will start, and the class size is 10, all 10 are open right now. Again, if you're interested, first send me an email, and then we can talk about how we can go about it. So for the first two, on this website, intro-to-bioinformatics.com, there is a form under, on the top there is a register button. If you type out that, if you click that, it takes you to a page which has similar information along with a link to a form. Please download that form, fill it out, and send it to me to reserve your spot for the first 2 courses. For the third course, just send me an email and I will send you a registration form, or because for that I need to have like a pre-screen with you to make sure that you are there and that way you would be able to handle the concepts covered in that course. All right? Does that make sense? Okay. And that's my email in the bottom. That's vthepar@partners.org. All right, so with that, I'd like to thank you for your time, and we can take questions now. I'm going to pause this video recording before I take questions.